Okay, good morning, everybody. I'm Robert Froemke from NYU, and it's my pleasure today to introduce Qiaojie Xiong from SUNY Stony Brook. Qiaojie was a PhD student with Min Lee at Hopkins, and then did her postdoc work with Tony Zader at Cold Spring Harbor. And her postdoc work for me was a revelation about plasticity and sort of the extended auditory system, and thinking about the behavioral relevance of changes in the corticostriatal system. She's had her lab now for several years at Stony Brook, where she continues to be a leader in the field on studies of auditory behavior in rodents, and thinking about sort of brain areas connected to kind of the auditory thalamic cortical system, including the TRN, and how arousal state might be modulated by glial processing related to the thalamic reticular nucleus, which is her topic for today's talk. So please take it away, Qiaojie. Sorry, one last thing. I'll be moderating the chat. Please type your questions into the Q&A, and we'll get to the questions at the end of the seminar.
Thank you, Rob, and good morning, everyone. It's my great pleasure to share my recent work here. And it's a quite new experience having a conference, a virtual conference, in replace with the SFN. So today I'm going to share with you a recent completed work in my lab in looking at how microglia modulate stable wakefulness through the thalamic reticular nucleus. Microglia has diverse functions in the central nervous system. They are the main immune defense system in the brain. In addition to that, they also participate in neurogenesis, synaptogenesis, myelination, vasculogenesis, and modulating the blood-brain barrier permeability. Accumulating evidence also indicated a potential correlation between microglia and sleep. First, in microglia, there are robust rhythms of circadian clock gene expression. Here showing some of the examples. Some circadian genes have their peak expression in the middle of light phase in microglia, and some has their peak expression during the transition between light to dark. In addition, also microglia displayed diurnal variation in morphology, and the microglia morphology are tightly linked with their function. So in the bottom left, I put in a graph showing the diverse microglia morphology from very ramified structure to amoeboid structure. The ramified structure are mainly correlated with their surveillance functions, and in response to the immune challenges, the microglia will retract their processes and become this unramified, and most of the time we call it activated structure. In normal brain, people have reported that the cortical microglia displayed diurnal variations in their from the unramified during the dark phase to the ramified during light phase. And that's in normal brain. So that together suggests there is a link between circadian and microglia function. Furthermore, people also reported that in mouse and rat models, sleep deprivation can activate microglia. This is microglia staining in hippocampus collected from control rat and sleep-deprived rat. And you can clearly see in sleep-deprived rat, the hippocampal microglia are largely activated. And similar findings were reported in sleep-restricted mouse. The chronic sleep-restricted mouse, their cortical microglia are largely activated compared to the control mouse cortical area. So that raises the question whether and how microglia regulate sleep. So that's the topic I'm going to share with you today. In this study, we investigated how microglia will affect wake and sleep in mouse. We first globally deplete microglia in the brain, from the mouse brain. To do that, we crossed the two transgenic lines of mouse, the CX3, CR1, CRE-ER. That's the transgenic mouse line that with tamoxifen induction, it will specifically express CRE in microglia. And then cross with the R26 IDTI mouse. In this way, we will induce the microglia, we will induce DT receptors specifically expressed in microglia. And then follow the DT injection, we can specifically kill the microglia in the brain. And this is showing an example of validation. Day 0 is before the DT injection. We did immunostaining for microglia, IBA1 as the microglia molecular marker. And by day 4, that's the first day after the 3 DT injections, we found in most of the tested brain regions, the microglia is gone. And then we performed EEG and EMG recording to analyze the sleep structure in these mice between before and after the microglia depletion. So this hypnogram showing one example recording from one mouse during daytime and nighttime, before in the upper panel, before and after the microglia depletion. So on the left, I labeled wake state, non-IEM, and IEM state. Because mouse mainly sleep during the day and being very active during the night. So we separate the analysis between daytime and nighttime. From the quantification data, and if you look at blue colored data here, you can clearly see during daytime, there's not much changes in the total duration of wake, non-IEM, and IEM sleep. And we also include a group of control mice in which they have the same genetic background, but instead of DT injection, they receive vehicle injection as a control. And in either group, we don't see much changes in daytime. But in nighttime, there is a clear reduction, significant reduction of total duration of this mice spent in wake. And correspondingly, there is a significant increase of non-IEM sleep, not much change in the IEM sleep. And then we further looking at the bout duration from individual vigilance states. In contrast to the increase of total duration, the bout duration of non-IEM sleep is showing no change. That means we only increase the frequency of non-IEM sleep, but didn't change the routine non-IEM sleep duration. And for the wake period, however, we saw the dramatic reduction of the bout duration. And then we further dissect the wake bout duration into quick arousal and stable wake. Quick arousal, we defined it as any wake period less than 1 minute. And we group them into quick arousal. And for stable awake, we defined any wake period with bout duration longer than 1 minute. And you can see here this in this unit, we are grouping in hours. So in control animals, highlight here in the light gray, this DT0, we can see there's in the dark phase, we can see there are several hours of wake period. But after the microglia depletion, we rarely see wake duration longer than 1 hour. There is a significant reduction of the long period wake time. And consistent with that, if we look at the transition numbers between wake to non-IM, non-IM to wake, there's a significant increase after the microglia depletion, but not much change between the IM sleep with other states. So together in this microglia global depletion experiment, we show that microglia depletion strongly affect stable wakefulness, and mainly through increasing the wake to non-IM transition. So next, we want to see what's the systematic factors that mediate this microglia depletion for regulating the stable wakefulness. So we did a metabolic metabolomic screening. To do that, we collect the brain tissues from sleeping mice and awake mice, and perform the screening to compare what are the metabolites that showing the strong difference in concentration between day and night. We total find 155 metabolites showing strong diurnal difference. And in panel B here, I only list a subset of them because that displayed the major family belong to the lipid family. And among the sphingolipid, many metabolites fall into this sphingolipid family. And in this family, we identified the ceramide, which is a bioactive metabolite that had been shown before can modulate neuronal activity. So we further dissect the different species of ceramide and compare their day and night concentrations. And for the C18, which is brain-enriched species of ceramide, there's a clearly difference between day and night concentration, with the nighttime with a lower concentration during night. And then we also test their diurnal changes in microglia-depleted mice, and we find in these mice, specifically for the ceramide concentration, their diurnal variation is gone. More specifically, there is an increase of nighttime ceramide concentration. So that kind of suggests that maybe ceramide can mediate the microglial interaction with the stable wakefulness. If ceramide is the mediator, then by manipulating the ceramide concentration at nighttime, we should be able to see the changes, the same changes that we observed in microglia-depleted mice. So to do that, we in a set of controlled mice, we inject camophor. This is a chemical to inhibit ceramidase function. So in this way, the camophor injection it will increase the ceramide concentration in the brain. That's in control mice. When we inject the camophor and perform the EEG, EMG recording, we found indeed it recapturates what we found in microglia-depleted mice. There is an increase of transition numbers between wake and non-IM, and non-IM to wake. I didn't put it here. There's also a reduction of the total wake duration and increase of total non-IM duration. And more specifically, I'm showing here, there's no effect in quick arousal about duration distribution, but there is a significant reduction of the long period stable wake. That's what we found in microglia-depleted animal. And then you can argue that this may be just in parallel pathway. It's unnecessary mediating the microglia modulation. So to test whether the ceramide is indeed mediated between microglia and stable wakefulness modulation, we test if we reduce the microglia concentration in the microglia-depleted mice, whether we can rescue the impaired stable wakefulness. So this is the same setting for the microglia depletion. After the DT injection to deplete microglia, we inject another drug called GW4869 into the mice. And this is this will inhibit the ceramide production. And in this way, we test whether that will rescue the stable wakefulness. And the quick answer is yes. You can see in the red circled data that's the mice receive DT injection and GW injection. Therefore, they have microglia depleted in their brain, but also have suppression of ceramide production in their brain. And in this mice, we don't see a significant change in their total duration of wake and no change in their stable wake about duration distribution. But in the control animals that they have microglia depleted in their brain, but no change in the no manipulation of ceramide production. Again, we saw a clear reduction of the long period stable wakefulness. So together, that kind of suggests microglia through manipulating the ceramide production will modulate the stable wakefulness. But how exactly that happened and whether this is a global homeostasis modulation or through a specific brain circuits, we next test whether there's a brain region that's strongly responsible for this microglia ceramide mediated effect. So first, we did a quick screening of microglia diurnal morphological changes in subcortical region. Here only showing a subset of screened brain regions that these brain regions have been all implicated in the sleep regulation. And among them, we found 5 brain regions that showing significant changes of microglia morphology between day and night. And then we test whether in these brain regions, the microglia are sensitive to ceramide concentrations. So then to acutely increase the ceramide concentrations in these brain regions, we inject ceramide through the cerebral ventricle injection, and then analyze the morphological changes of microglia in those brain regions. And among them, we identified thalamic reticular nucleus, which is a thin sheath structure surrounding the thalamus. And 98% of those neurons are pulvovalin positive. So in this area, the microglia quickly responds to the increase of ceramide concentration. We also confirmed this finding using specific transgenic mouse line called alkaline ceramidase 3 knockout mice. And in these mice, they have elevated ceramide concentration in the brain compared to the wild type mice. And we found again in these mice, specifically in TRN, the microglia are relatively more activated in the ASER3 knockout mice. So then that implicated that that suggests TRN may be the important brain region to mediate this microglia modulation to the stable wakefulness. To test that, we performed a local depletion of microglia by injecting the clotronate into the anterior part of TRN. The reason we chose the anterior part of TRN is because, first of all, it's a long spanning sheath surrounding the thalamus. And the injection is hard to target all the areas. And most importantly is because in 2014, there's a report from Matt Wilson's lab that the anterior part of TRN, which is more involved in limbic functions, they showed their activity are highly correlated with arousal. So then we picked this part of TRN and locally deplete microglia. And what we found is indeed, by locally deplete microglia in this anterior part of TRN, we found a strong reduction of total wake duration and strong increase in the non-IM duration. And consistently, there's an increase of transitions between wake to non-IM. And so that kind of putting together seems it's all consistent, indicate TRN maybe the important regions regulate stable wakefulness. And if that's true, we would expect to see the microglia depletion will affect TRN activity. Similarly, the application of ceramide application should be able to modulate the TRN neuronal activity. In these three panels, in summary, the answer is true. Yes, the TRN has a higher firing rate during night when the animal is awake. That's consistent with the previous report. And both external ceramide application and microglia depletion will decrease the TRN intrinsic excitability. So then next, we want to note how exactly TRN modulate the stable wakefulness. So we performed EEG, EMG recording combined with the tetra recording in the anterior TRN. As I mentioned before, the majority of TRN neurons are parvalbumin positive GABAergic neuron. And in the recording, we compared the TRN neuron firing rate between different vigilance states. And here showing a clear plot that TRN has much higher firing rate when the animal is awake compared to the animal is in non-IM sleep. And more specifically, when we aligned individual TRN neuron activity around the transition point between from wake to non-IM sleep, we find a sharp drop of their firing rate and slightly preceding the wake to non-IM transition. On the other hand, if we look at the transition from non-IM to wake, again, there is a sharp increase of the firing rate, but for most of the neurons, they increase their firing rate around the same time of the non-IM to wake transition. So this firing drop preceding the wake to non-IM transition motivate us to test whether the TRN neuron actually regulate or controls the wake to non-IM transition. So to test that, we use chemogenetic tools bidirectionally modulate the TRN neuronal activity. So in panel E, I'm sure. So in the upper panel, I'm just showing you the validation of the drug infusion. We are mainly targeting the anterior part of the TRN. So in panel E, we use the cell B cord system to chronically activate TRN neural activity and measure their effect in the sleep-wake structure. What's shown here is indeed by, cell B and cord to inhibit the TRN neural activity. So by chronically inhibit the TRN neural activity, we significantly increased the wake to non-IM transitions. And this is the control animal where we don't express SOB receptor, instead just a control gene. And on the other hand, if in another set of animal, we chronically increased the TRN neuronal activity, we can see opposite effect. By increasing the TRN neuronal activity, we suppress the wake to non-IM transition and similarly suppress the non-IM to wake transition. So then next we ask, so together here seems that TRN neuronal activity indeed can regulate wake to non-IM transition. Then what happened during the microglia depletion? What's the function of microglia modulated the TRN neuronal activity? So we then look at the TRN neuronal activity in microglia-depleted animal. And we use the same approach to deplete microglia in the brain and then measure the TRN neuronal activity. Or we find a significant reduction of mean firing rate in TRN, more significant in wake period. There is a trend of reduction in non-IM sleep period, but it's not significant. And for individual neural activity around the transition between wake to non-IM, we can see this is before the microglia depletion. Again, there's a lot of neurons showing strong activity and sharp reduction when it transits to the non-IM sleep. But in microglia depleted mice, we found very few neurons with the strong activity before the transition. There is still a stronger transition in wake compared to non-IM sleep, but the difference is much smaller. And you can see from here, the transition seems shallower between wake to non-IM. And similarly, from non-IM to wake transition, there's also a reduction of their firing rate difference. So then that suggests the microglia through regulating the ceramide concentration will regulate TRN neural activity, and that will in turn modulate the wake to non-IM transition, which eventually modulate the stable wakefulness. So if that's true, then we should be able to rescue the microglia depleted depletion induced impairment by activating the TRN neuron activity. So that's what we did in checking the time in the last experiment I want to show today. So again, we used the DREAD system to chronically activate TRN neuron activity. But this time, what I showed before is in control mice, is in wild-type mice. But this time we did that in the microglia-depleted animal. So, and then check the sleep structure. So what show here is in the control animal where we express just GFP and receiving the same CNO injection. Again, after microglia depletion, we saw the significant increase of transitions between the wake to non-IM. But with the HM3-DQ expressing animal, where we activate TRN neural activity, before and after the microglia depletion, there's no significant change in the wake to non-IM transition, and similarly to the non-IM to wake transition, and more clearly that in the GFP mice, again, we saw after the microglia depletion, we saw a reduction of the long period stable wakefulness. But in the DQ animals, we saw no significant changes in the stable wakefulness. That means by activating the TRN neural activity, we can rescue the impaired stable wakefulness from the microglia depletion. So I will end here. We show that the microglia depletion decreased the stable wakefulness during the nighttime, and ceramide can mediate this microglia modulation, and it's likely through the anterior TRN neuronal modulation. And this is our working model. During the wake period, the TRN neuron, there's lower concentration of ceramide in the environment, and there's a higher TRN neural firing rate. And during sleep, there's more ceramide and suppress the TRN neural activity, and the mice is mainly in the sleep. So I will end my talk here, and thank you for your attention. This is my lab and my collaborator, Xiaoyuki's lab, and my funding source. Thank you.
Okay, thank you, Qiaojie. Please type your questions into the Q&A box or in the chat. We have a question on if you've checked microglia status and TRN activity in artificially induced sleep with anesthesia. Are there similarities or differences between?
Oh, sorry. I was reading. Yeah, I was reading the question. Yeah. So, okay, yeah. So for we didn't record the TRN activity in anesthesia, but there were Yeah, I will answer this question in two parts. So the quick answer is we don't know what happened during anesthesia because we didn't perform that recording. We only once we mount the tetra on top of the mice head, we never actually put down the animal under any method of anesthesia. They are just freely sleep or wake by their own. So if I need to make a guess, I would think in general, the anesthesia is not the same as sleep. So the microglia, the TRN activity may or may not have the same phenotype between sleep and anesthesia. Why I'm saying that, there's a hint from the literature that microglial morphology, they have less ramified structure in sleep, but they have more ramified structure in anesthesia, as both compared to awake condition. So, so that's two directions from the wake. Compared to the wake condition, sleep, it seems that during sleep, it seems microglia are more active, but in anesthesia, the microglia are less active. So, so in that way, if putting together with our findings, I would guess the TRN activity will be more active in anesthesia compared to sleep.
So what's the action of ceramide? How does ceramide decrease excitability? You showed that both microglial depletion and ceramide augmentation decrease the F5 functions. What's going on there?
Yeah, so ceramide by external application, it will decrease the TRN neuronal activity. We are in the middle of testing in vivo the spontaneous firing rate, but in vitro, through the whole cell patch recording, there's a clearly inhibition effect.
Is that by opening potassium channels? What's the mechanism of the change in excitability? So
molecularly, there's some hint from the literature. It seems that there's a specific receptor on the membrane surface, but we haven't tested whether this is indeed the mechanism mediating in our system. Yeah.
And during, the normative operation of TRN, can you talk a little bit about how it is that you activate inhibitory cells? And even though you're upregulating this GABAergic system, you're waking animals up. Or conversely, if you then kind of shut it down, how do you lead to a drop in wakefulness?
Yeah, so so we are not sure the downstream. We're looking for the downstream target. One possibility is because naturally, like with the normal conditions, the TRN neuron, this particular anterior part of TRN neuron, has high firing rate during wake. So, so presumably they are inhibiting one of the downstream target, which release or disinhibit the modulation for sustained wakefulness. So our manipulation is trying to recapitulate what actually happened during the normal condition. But I'm not sure what exactly the downstream target yet. So the TRN receive input from hypothalamus. So I think, I don't have a direct evidence, but I think those extensive started the basal forebrain and hypothalamus wake and sleep modulation centers sending the signal to TRN. But from TRN mainly projected to the thalamus. But which nucleus that mediates this, we don't know yet.
Okay. Well, thank you very much. That was a great talk.
Thank you.
And we'll close the session. Bye, everybody.
Thank you.